Hey guys, my name is Max and welcome to Simple Biology. Today we're going to be talking about respiration. And respiration is actually one of the larger topics that we're going to be talking about in biology. And so this video is only going to cover the first part of respiration, which is what's called glycolysis. Now glycolysis is present in every living organism that we've come across to date. So obviously it's going to be important to understand. Now, before we get into how glycolysis works, we're going to review what redox reactions are. Now, this is a chemistry concept, but it's actually very simple. A redox reaction is one where electrons are exchanged between two or more compounds. So this colorful diagram or colorful formula right here kind of shows a generic redox reaction, which is this X compound has an electron and it transfers it to the Y compound, resulting in like this. So redox actually stands for oxidation reduction or reduction oxidation. Oxidation is the loss of electrons, whereas reduction is the gain of electrons. In other words, reduction will reduce the charge on that compound. So this is one example of a redox reaction. In this reaction you have sodium metal reacting with water to form sodium hydroxide and hydrogen gas. If you know about sodium hydroxide is actually an ionic compound. So this sodium is actually now an ion. So it has given up an electron and as a result has become oxidized, because it's lost the electron. The sodium metal lost the electron to become sodium ion in this compound. Similarly, the water, the H2O, was reduced to form hydrogen gas. And so that's kind of how a redox reaction works. Some other examples are rusting and burning. You may have heard rusting known as being oxidized. Same thing. Burning is going to be a little more relevant to what we learned today, in which I think that's burning, whether it's methane, whether it's wood, glucose, that's being oxidized. It's losing electrons to the oxygen that's also burning in that reaction. The important thing about redox reactions is that they allow electrons to fall to lower potential energy or lower free energy, depending on how you want to look at it. And what this does is it releases energy, usually in the form of heat or light or ATP in the case of respiration. Now, glycolysis is actually one example of a redox reaction. It's very similar to burning or combustion. In fact, glucose can burn, and in doing so it releases large amounts of energy all at once. The problem with this is that the cell can't control this energy. It's too sudden, it's too instantaneous. Instead of burning it all at once, it converts this energy in a stepwise manner. In other words, specific enzymes cause the glucose to break down in steps. By breaking down into steps and splitting this energy up into small increments, it allows the cell to harness it to produce its energy. Now, this little diagram shows combustion of glucose. This little square-looking thing represents glucose, and the electrons fall directly down to oxygen, releasing large amounts of heat, light, these different forms of energy. And, whereas in glycolysis, it'll step down in small increments until it reaches oxygen, and each one will allow the energy to be harnessed to do different things. Now, one example of these enzymes that's going to be especially prevalent when we talk about glycolysis is an enzyme known as dehydrogenase. Now, dehydrogenase takes 2 electrons and also 1 hydrogen nucleus, one is a proton, and takes all these at one time and transfers them to what's known as NAD+. NAD+ is the carrier that's used to transfer the electrons to what's called the electron transport chain. Now, the electron transport chain is basically this little section that takes the stepwise energy increments, and NAD plus in the form of NADH. It's called NADH when it has those electrons in that hydrogen. NADH transfers the electrons over to the electron transport chain in order for the electrons to fall one step at a time and for the energy to be harnessed. Now we're going to actually get into what happens in glycolysis. Essentially, the glucose goes through a series of different steps until it's broken into two separate sugars, and it keeps going until it reaches a form called pyruvate. You don't need to know all the intermediates, but there's just some general things you need to know. There are two phases of glycolysis. One phase in which energy is invested or added to that glucose, and one where the energy is released from the glucose. Now, if you can see, this shows kind of the different steps of glycolysis. You start with glucose. 2 ATP are used up to form the ADP and phosphate, and the phosphate will actually attach to the glucose and make the glucose higher energy. And then near the end, that energy that was stored in the glucose is used to produce 4 ATP, meaning a net gain of 2 ATP, and to produce 2 NADH. Again, NADH will later be used to produce energy, to produce more ATP in the electron transport chain. By the end of the process, glucose has split into 2 pyruvates and 2 water molecules. So as just kind of a net, you have 2 ATP gained, 2 NADH gained, and 2 pyruvate gained. Also, it's important to know that all of this occurs in the cytosol rather than the mitochondria, where the rest of respiration will occur. In addition, it's important to know that glycolysis also occurs in prokaryotes, in bacteria. Again, it's just in the cytosol, so that's where that happens. Also notice it takes no oxygen, so this can occur without oxygen. It's not aerobic. When we're gaining those 2 ATP in a net gain, or 4 ATP in right there, this is what's called substrate level phosphorylation. Now remember, phosphorylation is adding a phosphate group to another molecule. In this case, that other molecule is ADP in order to form ATP. Now this substrate level phosphorylation is a very small amount of energy relative to the total amount of energy that will be gained in respiration, but it's important to know that it's there, and that will come up later on. Lastly, we're going to look at how glycolysis is regulated. I mentioned this a little bit when we talked about enzymes, but there's one enzyme in glycolysis called phosphofructokinase, and it's regulated allosterically. If you want a review of what that means, go check out our enzymes video. But there are two molecules that play a role in regulating phosphofructokinase. ATP, which obviously you know it has a great role, it's what's produced by respiration, and AMP, which is adenosine monophosphate. If too much of the ATP is used, your cell will actually start using ADP to gain energy, and that'll produce AMP. So if there's AMP, that means there is a huge shortage of ATP, and so AMP will actually stimulate glycolysis and stimulate respiration as a whole in order to reproduce some of that ATP. On the other hand, if you have excess ATP, it'll slow down glycolysis in order to prevent too much ATP from building up. A third molecule, known as citrate, will also slow glycolysis, and citrate is a later form of glucose that we'll be looking at in the citric acid cycle. And so if there's excess citrate, that also means that the glycolysis is occurring too quickly, and so it'll slow down glycolysis. In addition to this, citrate helps to synchronize the effects of glycolysis and the citric acid cycle, which is the other half of respiration that we'll be looking at next. So glycolysis is just as simple as that.